Today we are very happy to host Eugenia Vazquez and Decio Eizirik. They are presenting differential immune and apoptosis-related gene signatures in pancreatic alpha and beta cells contribute to their fate in type 1 diabetes. A quick bio on Eugenia Martin Vazquez, she's PhD. She's a postdoctoral researcher at ULB. In Brussels, Belgium, she conducted her PhD in the Department of Integrated Pathophysiology and Therapy at the Andalusian Center for Molecular Biology and Regenerative Medicine in Spain, where she studied the pancreatic islet physiology during neonatal development and pathophysiology that characterizes type 1 diabetes. Dr. Martin Vazquez got her international PhD degree in 2023, then moved to Belgium to start her postdoctoral training within T1D research under the supervision of Dr. Decio Eizirik. Currently, she's a postdoc through Breakthrough T1D, and her research focuses on dissecting the molecular differences between pancreatic alpha and beta cells to find new approaches to protect beta cells in type 1 diabetes. Contributions from her PhD and her postdoctoral period were published in 13 full papers and reviews with approximately 170 citations. And Decio Eizirik, MD, PhD, really doesn't need too much of an introduction. He's very well known in this field. He's a professor at the ULB Center for Diabetes Research, and he has published over 430 full papers and reviews with an H index of 97. Pretty amazing. He's received many national and international prizes, and his research focuses on the mechanism of beta cell dysfunction and death in type 1 diabetes, on the dialogue between beta cells and the immune system, and on novel approaches to protect beta cells in type 1 diabetes. Thanks so much for joining us today. We're really excited to have you walk us through this paper.
So thank you very much for this kind introduction and for the opportunity to present our work. The paper and this talk is entitled Differential Immune and Apoptosis-Related Gene Signatures in Pancreatic Alpha and Beta Cells Contribute to Their Fate in Type 1 Diabetes. So, as you mentioned, this work was done at the ULB Center for Diabetes Research, where we work, but also in collaboration with the companies Inspero and Novo Nordisk. And I would like to highlight also the contribution of some key people to this study, starting by the pathogenesis of type 1 diabetes lab, especially Xiaoyan Yi, that is here. She performed all the bioinformatic analysis that we will show today. Priscilla Simath, that is here next to her. She performed part of the experimental validations together with me. And Sairo Jagurek, that he's a PhD student in our lab, but he works presently at Inspero. And all this work was done under the supervision of Professor Decio Eizirik. So here I left a QR code to access the preprint in case you are interested, and you can do a quick scan. So I don't need to explain what is type 1 diabetes, but there is a surprising feature of the disease by which the immune system is going to kill the pancreatic insulin-producing beta cells, but not the neighboring glucagon-producing alpha cells. So in non-diabetic conditions, we have our islet with beta cells, alpha cells, and in type 1 diabetes, in some cases, all the insulin-producing beta cells are gone. And this is surprising because both cell types, alpha and beta, are exposed to inflammation in type 1 diabetes, and both cell types are dysfunctional. So indeed, not only beta cells are dysfunctional, but alpha cell dysfunction is also a characteristic from type 1 diabetes. This is a paper from Marcela Brisova and colleagues, where they evaluated glucagon secretion in pancreatic islets from control individuals versus type 1 diabetic individuals, and they observed there is a decrease upon the disease. Together also with a decrease in the expression of alpha cell transcription factors, including MAFB and ARX. So the idea is that the extreme beta cell loss, together with the systemic hyperglycemia that characterizes the disease, is going to lead to a decrease in the expression of alpha cell nuclear factors, and therefore to impair glucagon secretion or to alpha cell dysfunction. However, in spite of this, as I mentioned, alpha cells remain in the islet during type 1 diabetes, and there is previous evidence of alpha cell resistance versus beta cell susceptibility in the disease. This is another paper from Laura Marocchi, that is a former member of our lab, And what the authors did at that time was to expose rat, pancreatic, and purified beta or alpha cells to the infection with the diabetogenic viruses, Coxsackie B5 and Coxsackie B4, and then evaluated apoptosis levels. So what they observe is that at a very low multiplicity of infection, beta cells display a very significant increase in apoptosis, while alpha cells remain happy in the presence of the virus until this multiplicity of infection was 4 times higher as the one compared to as the one needed to induce beta cell apoptosis. And even though here we have a significant increase in cell death, this increase is nothing as compared to the one observed for the beta cells. And similar results were obtained for CBB4, so rat alpha cells are more resistant than beta against Coxsackie virus infection. Then these resistant features were confirmed in human islet-like cells. This is another study from Florian Sinsak and colleagues, where what the authors did was to differentiate human-induced pluripotent stem cells into islet-like cells in vitro, and then expose these cells to the pro-inflammatory cytokine interferon alpha that is very important during early stages of the disease, and then analyze the gene expression by single-cell RNA-seq. So here we have the beta cells, interferon alpha treated and control conditions, and the same for alpha. And here the expression of selected genes. So I will just highlight some of them that are very interesting. So well, the color indicates the average expression, and the size of the circle, the percentage of cells expressing each gene. And for instance, we can see the expression of BCL2L1, that is an anti-apoptotic gene, is an inhibitor of the intrinsic apoptosis pathway that is strongly upregulated in alpha cells after interferon alpha exposure, but not in beta. And the same happens for HLA-E, that is an immune inhibitory molecule, HSPA5, that is a protective chaperone induced by endoplasmic reticulum stress, IFIH1, that is a viral sensor, and SOD2, that is a free radical scavenger mRNA. So all these protective genes are upregulated in alpha cells after interferon alpha treatment, but not in beta, that on their side are actually upregulating pro-apoptotic genes, such as ATF3 or DDIT3, that are not upregulated in alpha. So alpha cells show upregulation of anti-apoptotic, antiviral, and immune inhibitory genes, while beta cells upregulated ER stress, pro-apoptotic related genes. And then we also confirm these results, but in this case using native or primary human islet cells. So we analyze the expression of all these protective genes that I already described in alpha and beta cells from non-diabetic donors using single-cell RNA-seq datasets that are publicly available through the Human Pancreas Analysis Program. So in this case, we have non-diabetic conditions, and we can see that the expression of all these protective genes is upregulated in alpha cells compared to beta. So we have upregulation of protective genes in non-diabetic conditions and after interferon alpha exposure. So in view of this, our question is, why are alpha cells more resistant to diabetes-related damage than beta cells? And to try to shed some light into this question, we propose, or we develop our study following 3 different objectives or tasks. The first one is to compare the gene signatures of alpha and beta cells in different single-cell RNA-seq datasets in order to identify key genes that may be related to alpha cell survival or beta cell susceptibility based on their differential expression in alpha versus beta cells. Next, to confirm the findings in bulk RNA sequencing of alpha and beta cells. And finally, to define strategies to improve beta cell resistance based on the validation of the genes and pathways identified via modulating their expression and performing functional analysis. So in the context of the first objective or task, here you have an overview of the different single-cell RNA-seq data collections that we analyze. So we analyze data sets that, as I said, they are publicly available through the HPAP, and we focus on control or non-diabetic individuals for the majority of the analysis that we did. So we have a control cohort that is matching a type 1 diabetes cohort. So these individuals are young individuals. We have a second control cohort that in this case is matching a type 2 diabetes cohort. So these are older individuals. And then we have data sets from islet-like cells differentiated in vitro from stem cells. Again, focusing on control conditions, we have islet cells from stage 7 of the differentiation protocol. This is the last stage of the differentiation, so these cells are going to show a more mature phenotype, as compared to these other cells that we also analyzed from a different study, that they are in stage 6. So in a previous stage, they are more immature. So here you have the UMAP analysis for these 4 data sets, showing the alpha and the beta cell populations with a very clear separation between them. So for the HPAP, the younger and the older individuals, for the islet-like cells, stage 7 and stage 6. So there is a clear separation, as I said, based on the expression of canonical markers, glucagon for alpha cells and insulin for beta cells. So first thing we did was to perform a gene set enrichment analysis or pathway analysis in alpha versus beta cells across all the data sets. So here you can see this pathway analysis for the HPAP controls. And what we observed was that there was a consistent upregulation of immune-related pathways in alpha cells compared to beta. And this upregulation was conserved across all the data sets that we analyzed. So antigen processing and presentation, interleukin or interferon signaling were always appearing as upregulated in the alpha cells, suggesting that there is a conserved and enriched immune gene signature in the alpha cells compared to the beta cells. So in view of this, and in order to go a bit deeper into this immune-related gene signature, we perform an additional analysis of expression of selected genes in alpha versus beta cells, in this case only using the data set from the HPAP corresponding to the type 1 diabetes cohort. So we not only analyze the non-diabetic alpha and beta cells, but also the ones from autoantibody positive donors with a single autoantibody or with 2 or more, and from type 1 people with type 1 diabetes. So this for the alpha cells and for the beta. And the genes that we analyzed, we divided them into different groups. One that we call interferon-related and another one that we call defense-related genes. And what we observed was that there was indeed a preponderance for expression of these interferon-related genes in the alpha cells compared to the beta, but especially of these defense genes. As you can see, again, based on the color and on the size of the circles, these defense genes are much more upregulated in the alpha cells compared to the beta, and this is independent of the disease condition. So we confirm that there is an enriched immune gene signature in the alpha cells compared to the beta cells. Next, we perform a differential gene expression analysis in alpha versus beta cells. And again, we were able to pick all these immune-related genes in the alpha cells across data sets. They are highlighted here, HLA-A, B, B2M, and so on. And in this differential gene expression analysis, we observed that there was a gene that is here highlighted in red, MEC3, that was consistently upregulated, but in this case, in the beta cells. And again, this was conserved across data sets. So for the HPAP, we have MEC3 here and here. For the stem cell-derived islet-like cells, we were also seeing MEC3 upregulated in beta cells. So we analyzed the expression of MEC3 in alpha versus beta cells, and we found that it was not only strongly upregulated in the beta cells, but also its expression was almost undetectable in alpha. So given this strong difference in the expression and conserved across all the data sets in the expression of MEC3, we thought that indeed this gene could be a contributing factor to beta cell susceptibility to diabetes-related damage. So next, in the context of our second objective, that is to confirm the findings in bulk RNA sequencing, for this, we use a bulk RNA-seq dataset that is also provided by the HPAP. And in this case, these are FACS-purified alpha and beta cells from 12 non-diabetic human islet donors. So here you have the principal component analysis, clustering the alpha cells and the beta cells together. The differential gene expression analysis in alpha versus beta cells, showing the genes upregulated in beta cells in green, in alpha cells in red, and again MEC3 was detected here in the beta cells. And then the expression of MEC3 in alpha versus beta cells for the 12 donors, and we can see individual upregulations for all of them. So we confirm that MEC3 upregulation in the beta cells. And I forgot to say that the goal of performing this confirmation in bulk RNA-seq is that the single-cell RNA-seq analysis detect only a very limited number of events of differentially expressed genes. And with bulk RNA-seq, we can perform a deeper analysis. So it's always good to confirm the findings in bulk RNA-seq. So we confirm MEC3 upregulation in beta cells, but also the increased immune-related gene signature in alpha cells. And for that, we measure what we call the ISG, or interferon-stimulated gene score, that was significantly higher in the alpha cells compared to the beta. So just to clarify what is this ISG score was established from a previous RNA-seq. Here you have the reference to the paper from Alexandra Comans. And what we did was to generate a beta cell interferon-stimulated gene signature, or ISG score, which is based on the selection of 53 mRNAs that had more than a 3-fold upregulation following treatment of endocivita H1 cells with interferons, alpha or gamma. And the ISG score was calculated as the average expression of the selected 53 ISGs. So basically, it's another way to measure immune-related gene expression or upregulation. So with this, we confirm this upregulation in the alpha cells. And finally, in the context of our last task, is to define strategies to improve beta cell resistance, which is also the goal of this project in the end of the day, is to extrapolate the alpha cell resistant features to the beta cells. So we had to validate the genes that we identified. And the gene that we identified was MEC3. So I will explain a bit what is this MEC3, or maternally expressed 3 gene. This is a long non-coding RNA that is expressed in many tissues, including the brain, the placenta, the pancreas, of course. And its expression is lost in multiple cancers. And the reason is that MEC3 is a tumor suppressor. So as a tumor suppressor, it's going to induce apoptosis. Apoptosis, but also inflammatory responses, epithelial to mesenchymal transition, and endoplasmic reticulum stress via interaction with different proteins, for instance, with p53. So MEC3 has been widely studied in the context of cancer, but not in the context of type 1 diabetes. And this can be eventually a bit surprising, because in 2010, it was identified a small nucleotide polymorphism located within an intron of MEC3 that is strongly associated with type 1 diabetes susceptibility. And the role of MEC3 has been a bit studied in mouse models of autoimmune diabetes, of type 2 diabetes, but never in human islet-like cells. So we decided to perform the validations using this candidate in human pancreatic islet cell models in vitro. So we used the immortalized human pure beta cell line endocivita H1, and also primary human islets that were dissociated and reaggregated as human islet microtissues. And these experiments with the microtissues were done in the laboratories of Insfero, while the ones with endocyte cells were done in our laboratories in the UCDR. So they were performed in parallel. And what we did was to expose these islet cell models to pro-inflammatory cytokines in vitro. We use cytokine mixes that are characteristic of late or adaptive immune stages of type 1 diabetes, and therefore they are going to induce beta cell apoptosis. And we did this in the presence of a small interferon or short hairpin RNA targeting MEC3, so to knock down its expression. And then we perform apoptosis and gene and protein expression analysis. So we found that silencing MEC3 decreases cytokine-induced apoptosis in endocivita H1 cells. Here you can see the expression of MEC3 after inducing the knockdown using a small interfering RNA in gray bars, in untreated conditions, and in inflammatory conditions. We use interleukin-1 beta plus interferon gamma. So the knockdown of MEC3 was pretty efficient, between 50 and 60%, and the reduction in apoptosis in inflammatory conditions was confirmed by two independent methods, Hodge propidium iodide staining and fluorescence microscopy, and also by analyzing caspase 3,7 activity. Similar results were obtained for the human islet microtissues. In this case, the knockdown was achieved using a short hairpin RNA, and the inflammatory conditions were slightly different. We included in the mix TNF-alpha and interferon alpha. And again, we confirm a reduction in apoptosis by analyzing caspase 3,7 activity. So here you may wonder why we are using different inflammatory conditions for these two models, since it might be a bit difficult to compare between the two. Also because for endocyte cells, this inflammatory exposure was only for 48 hours, while for the microtissues, we expose them to 6 days of cytokine treatment. So we perform additional experiments by which we expose endocyte cells to the usual cocktail of cytokines that we use for this cell type, interferon gamma plus interleukin-1 beta, and also to the 4 cytokines together. So we confirm the knockdown in any of the conditions, and then we analyze caspase 3,7 activity after 48 hours of any cytokine mix, and also after 6 days of any cytokine mix. So we were able to confirm that the silencing also protects endocyte cells after 6 days exposure to the same conditions that we use for the human islet microtissues. We also found that silencing MEC3 decreases interferon gamma-mediated STAT1 signaling in endocivita H1 cells. So Western blot analysis performed in cells silenced for MEC3 and exposed to interferon gamma at different time points showed that the levels of phosphorylated STAT1 versus total STAT1 were reduced after the silencing, meaning that the activation of this pathway was reduced. Here you have the area under the curve. So probably as a consequence, as a functional consequence of this downregulation of the STAT1 signaling pathway, we found that silencing MEC3 also decreases the expression and the secretion of the pro-inflammatory chemokine CXCL10 in endocivita H1 cells. Then we found that silencing MEC3 decreases also cytokine-induced HLA class 1 expression. In this case, these are the human islet microtissues. These experiments were done by 3D staining and microscopy. And what we found was that the strong upregulation of HLA class 1 protein expression induced by the pro-inflammatory cytokines was mildly but significantly reduced after the silencing of MEC3. And besides, we performed the staining with a beta cell marker that is NKX 6.1, and we found that the silencing of MEC3 increases the beta cell fraction in inflammatory conditions in the human islet microtissues, as shown by the increase in NKX 6.1 positive cells. So these are the main results from our study, and the conclusions that we reach are first, that alpha cells have increased interferon-related gene expression than beta cells. Together with the higher expression of anti-apoptotic and free radical scavenger genes, these results suggest that alpha cells are better equipped to endure viral and immune-mediated stress than beta cells. Second, MEC3 has higher expression in beta versus alpha cells across the different human single-cell RNA-seq datasets studied, and the depletion of this gene partially protects beta or islet cells from cytokine-induced damage and apoptosis. And finally, combining large-scale single-cell analysis indicates key features underlying alpha cell resistance and beta cell susceptibility to diabetes-related damage. And this data identified the long non-coding RNA MEC3 as a contributing factor for beta cell susceptibility to immune-mediated damage. And that's it. I would like to thank all the people from the UCDR, the funding agencies, and all of you for your attention. Thank you.
Thanks very much, Eugenia. Great talk. Very interesting findings. Hoping to open it up to people in the audience. We have a large audience, so.
Should I stop sharing?
I'm sure, please.
Hi, can I ask a question?
Yes. Hi, Ernesto.
Hi, how are you?
Good.
Nice talking, Eugenia. My question is, have you looked at upstream, like the receptors? Because I imagine the alpha cells and beta cells, they respond different because they have something different upstream in the receptors, maybe like the downstream, like the signaling. Have you looked at those?
I am not 100% sure, but I think in terms of gene expression, they are pretty similar in terms of receptor expression. Actually, we are starting to look into this in a bulk RNA-seq that we have generated from alpha and beta cells differentiated from iPS. And I don't remember very strong differences in the expression of the receptors for cytokines and so on.
And just to add, the receptor for the Coxsackie virus is the same in both. The virus enters the alpha cell and is kicked out, while in beta cells it proliferates and eventually kills.
It could be also some accessory proteins, right? Because some of these signaling pathways, they have some accessory proteins that they drive the signal in one way or the other.
The point is that alpha cells are more resistant to virus, to palmitate, to ER stress. So it's several mechanisms, and it's difficult to understand what is the evolutionary logic, because beta cells are more important.
I had a question for Eugenia, or both of you, Decio too. I noticed like in the one slide she presented, HLA-E is enriched in alpha versus beta plus interference. What can you say about the mechanism regarding the NK axis there? Right, because HLA-E is involved in sort of establishing and maintaining that NK access, good NK cells versus bad NK cells. I mean, could there be something there?
That's a very good question. Actually, I never thought much about NK cells.
Yeah, they were hot for a little while back, and then they kind of dropped down a bit. But
I think this is something that Roberto Malone is looking at.
yeah.
So this could indeed be a mechanism of protection, because HLA-E not only protects against natural killer, but against CD8, which is also important. And one point that Roberto has, we had a review together a couple of years ago, is that the insulin granule has zinc and the alpha cell granule does not. And zinc is an accessory molecule, for instance, in vaccines. So, well, this is speculative at that moment, but it's possible that the insulin granule is more immunogenic than the alpha cell granule. One thing I see that Maki Nakayama is there, and Maki has been looking at these cells that recognize islet peptides. Maki, if you are listening, did you find these cells that recognize fragments of glucagon?
No, thank you for asking. We have analyzed TCRs from, I think, at least 7 donors and tested all glucagon reactive TCRs, but we have never found those ones. But regarding HLA-E, we found one TCR that can respond to peptide presented by HLA-E, but it was not islet peptides. It's more like the viral specific peptide presented by HLA-E. So I don't know, it is protective or more like destructive, but you are definitely right. CD8 T cells sometimes use HLA-E to interact with targets.
Yeah, I mean, it's interesting. Here's a question from a couple questions. Patty Gerder, Medical College of Wisconsin. Thank you. Very nice work. I wanted to know if there's an effect of MEG3 on beta cell functionality. What will happen if MEG3 expression increases in alpha cells? Does that make the alpha cell prone to immune destruction?
Yes, so we have analyzed glucose-stimulated insulin secretion in the human islet microtissues after MEG3 knockdown, and we saw that there is an increase in the insulin secretion in inflammatory conditions. Also a decrease in standard conditions or in untreated conditions that it's a bit difficult to explain because we do not observe any detrimental effect in untreated conditions, but this is what we got, so it's what it is. And regarding MEG3 expression increase in eventually this is something that may happen, that the upregulation of MEG3 in alpha cells may render them more susceptible to cytokines, but this we haven't done. It would be a good indirect confirmation of the protective effects of the knockdown, but since our primary goal is to protect beta cells, we stop there. But yeah, this is something that we keep in mind.
Yeah. David Hill, does knockdown of MEG3 in the beta cells change their functional phenotype at all apart from response to cytokines?
Yeah, so basically the same. The function we analyze, we measure the expression. Also because there are papers about MEG3 but in mouse models, as I mentioned, for autoimmune diabetes and type 2 diabetes. And for type 2 diabetes, for example, the expression of MEG3 is decreased. And the decrease in the expression is linked to a detrimental phenotype in terms of function. So a decrease in beta cell identity genes, a decrease in the function. So this is why we tested glucose-stimulated insulin secretion. We measure expression of different genes in endocyte cells like MAFE, GLIS3, insulin, PDX1, and we saw no major impact.
Okay, thank you. There's here's one question from Yuval Dor. Delta cells are preserved in type 1 diabetes. Do they share features with the alpha cells, for example, resistance to stress and cytokines?
That's a very good question. For the moment, we haven't gone very deep into the analysis of delta cells, but I think this is also an upcoming task that we will do, because it's true, delta cells are also there in type 1 diabetes. But we haven't done a gene expression analysis comparing also the delta cells.
Yeah, just getting started. There's a lot more to do. I had another question, kind of off the side question, but if you look at some of these, like how MEG3 is driven, a lot of times in these different cell in vivo structure or systems, you've got like hypoxia as a driver for MEG3. Like if you look in the heart, MEG3 gets induced in cardiomyocytes after like a heart attack or MI. In the kidney, if they have a hypoxic injury in those cells, they get an increase in MEG3, pulmonary artery happens. So like, could you think about the fact that maybe beta cells are inherently dysfunctional, right? And then they are hypoxic. and that could be driving MEG3. Could that be part of the cytokine response or no?
I don't have much knowledge about that to do a big comment. I don't know if Decio maybe.
It's an interesting question. We have studied before, but this was in rat beta cells, and cytokines induce some of the genes related to hypoxia recognition. So for a while, we thought that cytokines, as you suggest, Monica, were inducing an hypoxia-like situation. However, when we moved to human islets, the effect was not so clear. And I think a big difference between rats and humans in beta cells is that in the rat, the cytokines inhibit in an important way the Krebs cycle. So it creates an hypoxia-like situation. In humans, is much less. It's and we never really explained this. For instance, nitric oxide is crucial in rat-induced beta cell death and is irrelevant in human islets. For instance, if you block nitric oxide formation, you prevent Krebs cycle dysfunction in rat and you preserve them. In human, it doesn't make any difference.
Thank you. All right, we just have time for one more. Let's see if there's anything. Theodore.
I think Maki also has her.
Maki, you wanna, or Theo?
Can I?
Or Maki? Maki, go ahead.
Yes, thank you. Very beautiful story. Thank you. Thank you for your presentation. So I have a very quick question. Have you ever tested if the MEG3 expression levels are different between like is it age associated, like younger babies have higher or lower MEG3 expressions?
Yes, so the analysis we did is in these two data sets from younger and older individuals, and we didn't see any difference in terms of MEG3 expression or in terms of, in general, global gene expression. So there is no difference in this analysis that we did caused by age or BMI, that were the two parameters different between the two cohorts. We kept them separated because they were provided like this by the age PAP, but no, we didn't see any age-related difference. But it's true, we didn't go deeper into maybe analysis at a very, very early ages or whatsoever.
Thank you.
Yeah, you had some good data there with the 1 and 2 autoantibodies, distinguishing the changes, some of the early changes. So maybe you'll have to slice into that, and as you get more time and more information, it'll be kind of, you'll illustrate those differences. Here's something from Theo Dos Santos. For the expression of the defense genes in the T1D and autoantibody positive donor alpha cells, while there's an increase in the expression of these markers, the percent of alpha cells where these markers are detected can still be low. Could we assume that there may be some alpha cells that are actually susceptible to destruction, which enriches the alpha cell survivors in the T1D islet? That's question 1.
Yeah, I think this is a possibility, of course. Alpha cells are dysfunctional and they are showing eventually similar gene expression signatures as beta cells, besides these differences. So it might be, of course, that some alpha cells will die.
And then the second question is regarding the points that Dr. Eizirik and Dr. Nakayama brought up. Is the lack of TCRs against glucagon fragments influenced by L cells exposing glucagon-like fragments to the gut-associated lymphoid tissue, which then contributes to the tolerance for glucagon? Sorry, my mic wasn't connected, he said. Yeah, that's a really interesting question.
According to Roberto, this could be a factor of protection because immune cells are to some extent educated in the gut.
All right. Yeah, it looks like
And I ask one question, coming back to what Teodoro dos Santos say, because you just published this very nice paper in JCI, and you did electrophysiology in alpha cells from controls and people with type 1. Did you see any heterogeneity in the alpha cells you studied? Because you study quite a lot of alpha cells.
Theo,
Yes.
oh, thank
I
you.
I got my
Hi.
mic. Sorry about that. I'm not exactly
That's
at my home station, and I was digging around for a camera with a mic.
no worries.
Um, there is a tremendous amount of heterogeneity in both the non-diabetes alpha cell cohort and also the alpha cell cohort that comes from donors that had type 1, which in our paper was more established type 1. It's never cut and dry. There's a considerable amount of overlap between the two cohorts as well. It's just that on average, or the median values for the type 1 alpha cells show dysfunctional electrophysiology, which do show up as statistically significant. But there are a number of alpha cells that if you took them out of the cohort, you wouldn't be able to differentiate which cohort they came from. So there is heterogeneity in the electrophysiology and the functional behavior of these alpha cells, even in long-standing, and we're talking multi-year type 1 donors, for sure.
Because coming back to your question, I think this is very interesting information, because it seems that the alpha cells don't die. Now. The mass of alpha cells, even in long-term type 1, is preserved. So either some died and there was proliferation, which is very limited even in alpha cells, or the heterogeneity is more at the level of function than at the level of survival. And I don't think that function and survival are always synonymous.
Yeah, no, I agree with that. It's a very good point.
Are you guys seeing survivor alpha cells clustering away from these immune dense regions on the way to type 1 or at diagnosis?
Perhaps Maki know, we don't know. You are mute, Maki.
Maki?
Sorry, could you repeat your question?
The
Sorry.
question is whether when T cells are invading, when you see insulitis, if the alpha cells are more far removed from the T cells than the beta. I don't know if anybody has looked at it. It's an interesting point.
The survivor alpha cells, like for Theo's work, the ones that are still electrophysiologically functional, or even just, have functional markers in histological sections. I just wondered if they are away from these immune dense regions, but yeah, to be continued.
Yeah.
Right, yeah, need more data. So any last questions, last call for questions? Oh yes, Vera Kravitz did mention in her study that in rodents, alpha cells act like immune recruiting hotspots. That's right, she did have that. She did have that finding, I remember that. So maybe we're gonna have to get her here back again along with Roberto Malone. So for the next one, but thank you all for coming. Great finding and presentation, Eugenia, and thanks all for the great discussion. Look forward to the next one.